Not joining us.
Inouye.
No way. No way. Is that what it is?
Yes. You can spell
Okay. Right. Thanks, Lindsay. Very pleased to introduce Mike Inouye, who is from Cambridge, from this public health and primary care, and works on also population genetics, and he's going to tell us about that. Right.
Thanks, George.
And thanks everyone for making the time, including our colleagues online. So I've got a fairly full slide deck that covers a lot of ground because I wasn't quite sure what people would be interested in. So we'll just sort of go through it. But if you want me to go into depth on anything, maybe the best thing would be like afterwards, just come on up and we can go through it. So I'll tell you a little bit about our work in what I'm calling systems genomics, or AKA sort of multi-omics, that sort of thing, and how we can apply it to populations. And that's really been a focal point of, if the slides will go, no. Left, right. How about the click?
There must be someone more technically in depth than me.
Okay, clicking works.
Everyone is more technical. Oh, it works.
Yeah, the mouse works. Okay. Great. So I can't go backwards, so we're only going to move forward. So our lab really focuses on this area of systems genomics, and what I mean by that is we spent a lot of time doing genome-wide association studies, but now we're really interested in how we can actually start combining genetic variants and their corresponding effects to construct these things that are called polygenic scores, which many of you work on here. And how we can use those polygenic scores to predict and ultimately prevent or manage common diseases, with a particular focus on cardiovascular and metabolic diseases. The systems part is really trying to, how do we incorporate other molecular and anthropometric and that sort of data as part of sort of a multi-omic framework to try and understand the etiology of common diseases. And to identify potential targets for therapies. And really an offshoot of all of that is that we also love to package and make available broadly, fairly, a lot of computational tools and resources, so software, online portals, catalogs, dashboards, that sort of a thing. And by fair, we're very much adherent to this concept of findability, accessibility, interoperability, and reusability. So all the stuff we do is open source, widely available to the community, and we try and be as open as humanly possible. And we depend on a lot of really excellent underpinning infrastructures, which I've listed there. So why do we focus on systems genomics? Well, some of you are probably familiar with this now relatively old schematic from Civilek and Jake Lucis at UCLA. So they did a lot of conceptual sort of groundwork in this area, trying to articulate what systems genomics is and why it's sort of useful. And really what they came up with is that you're trying to integrate a lot of this really high dimensional multimodal data for humans in particular, but also for some model organisms like mice, to try and understand the effects of naturally occurring genetic variation and how those sorts of changes sort of propagate their effects through different, particularly molecular systems. So the transcriptomes, the proteomes, the metabolomes, microbiomes, et cetera. So the idea is if you integrate all this data together, you can try and you not only can sort of create things like prediction models, but you can also try and trace the sort of causal paths of those effects through these different layers. And we've spent a lot of time trying to dissect each one of these layers. So whether it's the genetics, the proteins, the metabolites, the microbiome, and we've been looking at not just within these, but also across them as well, and trying to integrate across multiple layers. So that's really a focus of our lab, and our sort of core sort of goal, right, is to create these prediction models and to understand etiology. So I'll spend a bit of time. I'll start with polygenic scores and sort of build my way up. So many of you are probably familiar with this already. Well, we don't need to go over it, but the basic background, right, is that we've done a whole bunch of genome-wide association studies. We have a whole bunch of both individual level and summary statistic data, and we can now construct these scores out of these genetic sort of SNP to phenotype associations that we've found, right? And this sort of single number, this polygenic score that you can calculate for each individual, gives you a sense of the sort of genetic susceptibility or predisposition to a particular disease or trait. And you can plot these scores for a particular population, and they generally follow Gaussian distributions, where some people are at low risk and some people are at high risk, but most of us sort of follow it in the middle of that distribution. And there's a bunch of potential benefits to polygenic scores, which it's worth articulating. I'll go over risks a bit later on. So one is obviously in disease risk prediction, very much in the same sort of way as systolic blood pressure or cholesterol, et cetera. And frequently, I would say essentially, it makes no sense not to. We combine these polygenic scores with other sort of risk predictors, right? The blood pressures, the cholesterols, age, sex, et cetera, to create these sort of integrated risk models. You can also use polygenic scores to help improve and refine diagnoses. And by that, I mean things like the difficulty in separating type 1 versus type 2 diabetes for adults. So that's been demonstrated by, I think, some people just down the road in Exeter, if I'm not mistaken, Andrew Hattersley and co. You can also use polygenic scores for how we might be able to slow disease progression and recurrence. So how do we better sort of target statins, for example? How do we improve? You can also use them for improving screening. So, for example, Brent Richards has done some good work on this, like in osteoporosis. You can potentially also use them to prompt risk-reducing behaviors. Obviously, very much a difficult area, but there are some studies ongoing that are trying to demonstrate that if you give people their sort of polygenic risk for a disease, that they then do some sort of behavioral response to that, whether it's losing weight or engaging health coaches, that sort of thing. And there's some evidence that that might be the case, but there's again a lot of evidence that there's not. And also, you potentially could use polygenic score to help improve the cost and efficiency of clinical trials. For example, by enriching for your outcome of interest by selecting people at high polygenic risk, and also and correspondingly, you can also potentially reduce the size of your trial you might have to run because you're able to target a particular group more efficiently. So we've probably been in the polygenic score game since 2010, I think. So there's a whole bunch of stuff that I'm not going to cover, but I'll just cover the kind of the cardiovascular component of it. And that really began circa sort of 2014 or so. So this sort of study took us a while, but back when we had really just sort of early sort of cardiogram plus C4D summary statistics available to us, these large genome-wide meta-analyses, we were able to use really primitive methods like LD pruning to construct polygenic scores that were maybe a few hundred or a few thousand SNPs depending on what array you were looking at. So we did some early work in this. We constructed a 49,000 SNP polygenic score. We looked at it in a bunch of external cohorts, whether that was the WT-CCC or a familial hypercholesterolemia cohort like Argos. But in particular, we were interested in these prospective population-based studies like FinRisk or the Framingham Heart Study. And really what we found, even back then, was that when you add this still sort of primitive polygenic score to conventional risk factors, which I named before, age, sex, cholesterol, blood pressure, et cetera, you could get pretty significant odds ratios, obviously, for the PRS, but also improvements in net reclassification. So according to some of these numbers here, and that was the case for both incident cases and for controls. And one of the nice things that we could do even at that point was take a perspective of well, this is germline genetics, we don't have to assume that it's only measurable at baseline, that, this information, could be available throughout the life course of the individual. So, can we actually construct these things like start looking at things like lifetime risk? So we constructed these sort of original sort of inverted Kaplan-Meier curves, which you might have seen sort of a lot all over the place, really. And sort of demonstrated that, depending on what sort of quantile of the polygenic score you're on these very different sort of trajectories of risk over time. To the extent that, if you're in the top 20% of a polygenic score, on average, you'd have an event, probably 15 to 20 years before someone who was in, say, the lowest, sort of most protected 20% of the polygenic score. That's roughly what you see here for the thin risk males. So that was nice. We constructed a score. We demonstrated, how it could, its sort of validity in observational data. That was done in collaboration with our colleagues in Finland. They then took this polygenic score forward into what really needed to be done, which was a kind of a prospective study looking at how people responded to this information. So did they accept it? Did they find it useful? What did they do in response to it? So that's what the point of this gene risk study was, which is run by Elizabeth Whidden and Samali Rapati. It's published last year. As part of that, they really did something cool, which is they put together a new sort of risk visualization tool called Cardiocompassy. And that sort of made available an interactive method for individuals in the gene risk study to sort of play with their conventional risk factors. So what would their risk be if they lowered their blood pressure by 10 millimeters of mercury? What would it be if they stopped smoking, et cetera? So they could sort of play with this and they could see it in a personalized fashion, right? Where they actually had their polygenic risk and they could actually look at those Kaplan-Meier curves and how they went up or down as according to what sort of behaviors they might be exploring. So in response to this information, they actually found some pretty positive things, which sort of allayed a lot of concerns, particularly at the time, around how anxious people got, whether they found the information useful, et cetera, et cetera. But they also showed that that there was an association between the polygenic score and whether people sought a physician, whether they lost weight, whether they signed up for health coaching. They didn't see any relationship with smoking cessation. And so while this is not a trial, because everyone got their polygenic score, it does sort of give some evidence that, that there could be some positive effect of this information for the individual. And there's a lot of proper randomized controlled trials now ongoing, particularly in the US, trying to look at this topic. This is kind of a theme that I'll return to over and over, the need for translational studies, including pragmatic trials. So we continue to take this sort of area forward and try to look at more sophisticated methods for constructing polygenic scores using much larger data. So way back when UK Biobank genotypes first came out, we immediately started working on constructing a new polygenic score for heart disease. And so we published that in 2018. We worked on then another major component of cardiovascular disease more broadly, which is stroke. In particular, we focused on ischemic stroke, so we constructed the polygenic score for that. And really what we were aiming to do as we broadened our sort of work in the CVD area was we wanted to bring the various sort of polygenic scores for these different modalities together into a joint model with conventional risk factors so that we could actually look at cardiovascular disease as its own sort of outcome, right? Because that's currently what the guidelines use. So we're not in this space. I don't know if there's any cardiovascular aficionados here, but it used to be that you'd focus just on coronary heart disease with the original sort of Framingham risk score. But more recently, particularly, I think because the treatments are essentially the same, they started combining a lot of the outcomes into this more heterogeneous cardiovascular disease, which encompass both heart disease, stroke, and a few other things. So in a couple years ago now, actually, we put these two polygenic scores together, dropped them into the same risk equation as those conventional risk factors, and tried to estimate what the sort of public health impact would be if you had a polygenic score or these two polygenic scores measured alongside those conventional risk factors in UK Biobank. And this was kind of nice in that one of the main, it was one of the sort of first studies of that variety, but it also importantly, we tried really hard to correct for the healthy participant bias that exists in the UK Biobank. So working together with colleagues at the cardiovascular epidemiology unit, particularly Lisa Pennells and Stephen Kaptogi, we were able to use some of their work in they had sort of baseline CVD risk estimates, agents X stratified for from the CPRD database, which many people here probably know is primary care health records. We could then use those baseline estimates to recalibrate the risks in UK Biobank so that they more accurately reflected a primary care population. So we ended up, I think at least, with more sort of realistic estimates of risk reclassification. And when we did that, the sort of our performance, our sort of anticipated performance went down. Fine, whatever, it's more realistic. But in the end, it still actually wasn't that bad. In that we estimated that if you actually dropped in PRS alongside these other risk factors, that you could increase the number of CVD events prevented per 100,000 individuals by about 7%, or in a sort of increasing number of prevented events by about 72 per 100,000 per 10 years. And that's keeping the number needed to treat right about the same. You can run a quick, just to give a more sort of firm view of what this actually means. If you extrapolate to like UK wide for all 40 to 79-year-olds using ONS sort of demographic data, that sort of gives you roughly 20,000, an increase of about 20,000 CVD events prevented per 10 years. If you were to roll this out UK wide, which isn't realistic, but it gives you a sense of like how many events we're talking about here or could talk about. So that sort of piqued our interest in well, what can you actually use a polygenic score for? And one of the things that that our unit was interested in is CVD risk screening. So so our colleagues Ryan Chung and Angela Wood sort of asked the question, what role could polygenic scores play for prioritizing people for formal cardiovascular risk assessment? And to do that, they again used UK Biobank, but focused on the primary care health records, and essentially used that as one of the approaches that one could use to prioritize people for a formal assessment, which was taken as the UK Biobank baseline measurements. So they could have compared up the relative usefulness of measuring the same two PRSs in the primary care population individually and in combination with the primary care records as an approach for prioritization for formal assessment. And when you do that, you find that if you start with I guess this graph's unfortunately a little bit small, but you frequently see the PRS and the primary care records giving similar numbers of in terms of NNS, or number needed to screen in order to prevent one event, as well as the number needed to invite. But in the end, what you see is that the best sort of approach is to combine them, where if you were to use, well, age and sex specific prioritization thresholds for false negative rate, about 5%. Primary care, if you use primary care records alone and the Q risk factors from them, you get an NNS of about 149 for males and about 280 for females. If you were to layer PRS on top of that, you'd see a reduction in NNS from 149 to 116 for men, and a reduction from 280 to 180 for women. And these were significant differences. So that gave us, some sort of positive evidence that PRSs could potentially be used as a way of prioritizing people for formal screening in cardiovascular disease. So these are all kind of good sort of thought experiments, because we're in this space of having these new predictors, which we can calculate relatively early in life, but not really knowing how to use them. So you go through this sort of mental exercise of reimagining what screening could be if you had something like this. And indeed, we're not the only ones. There are colleagues here who do it, who are in the polygenic score space. There are hundreds and hundreds of people around the world working in this space, and many of whom are doing similar things. But one of the things I think we really need is this sort of, clinical workflow for polygenic scores. And our colleagues in the US, particularly out of Mass General, have been working a lot on this together with the Veterans Administration, or the kind of the after military part of the US military. And they've sort of been constructing this way of doing a clinical workflow, of generating the data, calculating the PRSs, generating a clinical report, that sort of a thing. That was sort of published, some of the basics of it in 2022, and they showed they could validate it on their Mass General Brigham biobank, and they could actually start running clinical assays for some of the veterans who show up to their VA. This just came out like yesterday, so I had to whack it in here because it's pretty cool. I actually haven't read the whole paper yet, but it's worth a read, I think, where they're talking about implementing pragmatic trials of polygenic scores within the VA. And then they're looking at primary outcomes like time to diagnosis of some headline diseases, including some of the ones that we're interested in, right? Like CAD, diabetes, AFib, several cancers, right? And it looks like it's pretty systematized. So it's worth having a read, but this was kind of like the thing that I was really hoping to end with and be like, we need pragmatic trials. And it's like the paper comes out like hours before you're talking. You're just like, I got to put it in. So here it is. Anyways, if you don't listen to my talk, read this paper. Okay, so polygenic scores, all great. There's lots of potential benefits to them, but there's also a lot of risks. And particularly being in a public health department, you don't sort of put your blinders on like you would if you were just a geneticist and say, look, I've got this thing, it works. Of course it works. Why wouldn't it work? There are downsides, right? And of course, there are the typical ones, like for any risk factor, right? So in many senses, sort of polygenic scores aren't special. So false positive results, right? You can have, it can be a relatively imperfect, predictor. You might incorrectly classify people as being at risk when they're actually not. There are issues around failing to convey the uncertainty here on the individual estimate, which is also the case with other risk factors, including blood pressure, cholesterol, et cetera. But in the case of genetics, you do have this added sort of, gasoline on the fire sort of thing of genetic determinism, which is a major source of confusion for the public, right? Because they hear something that's in your genes, it's genetic. They think that it's predetermined and that essentially, if you got it, you're going to get the thing, right? So polygenic scores kind of play right at the interface of that, of being genetic, but being a risk factor. So we have to be very careful about how we deal with genetics and how we talk about and perform studies with polygenic scores so that we don't reinforce genetic determinism. If you talk to the public, they don't have the tools to separate these two things. You really need to be careful about this. Anyways, biases of representation. These are things like if you have a risk factor and it performs really well in the population that you discovered it in, it's kind of why you discovered it. it might not work so well in other populations. So if you go into another healthcare system, if you go into another country, another ethnicity, another ancestry, your risk factor doesn't work quite as well. And that's certainly the case with polygenic scores. And I'll talk a little bit more about that. Healthcare professionals, there's a whole nother thing. It's not necessarily like another protein that a healthcare professional can run, in their lab. They need to have some sort of training and communication framework for genetics. And a lot of healthcare professionals don't have that training, or don't have it to a sufficient level where they can sort of have a kind of an appropriate conversation about genetics with their patient. So there is an aspect of training there. And that feeds directly into communication of results, which feeds directly into potential increased risks for psychosocial sort of things like anxiety, depression, etc. And people do get these things, and we're really just getting a sense of what the rates are. Thankfully, and in some of the studies I've talked about here, is like whether it's gene risk or actually whether it's the genomic PLC acceptability study, the heart study, they don't find much anxiety. They don't find sort of much really poor psychosocial effects, which is good, right? So it's not something that a wide swath of the population is going to experience, but we need to better understand, the rates of these things and what people like, more sort of qualitative research around, understanding why that is for some people and not others. And a lot of this work has been done by the Polygenic Score Task Force, which is part of the International Common Disease Alliance. And that's something that Kristen Willer and I co-chaired back when. And it was a really great sort of collaboration, which I'll talk a little bit more. There are also societal risks, which need to be sort of acknowledged and in some cases discussed, right? Many people here have probably seen some high-profile media around polygenic screening for human embryos. You can calculate polygenic scores for embryos. You can look at educational attainment. You can look at coronary heart disease risk. You can look at breast cancer, all this stuff, right? And then, well, if you paid for it, you can then make choices about whether you go with that embryo or not, right? That's a big box of yuck that we're not going to deal with, but it's an ethical minefield, and depending on who you talk to, it has no validity anyway. So why talk about it? But still, it's something that is being pushed by companies, and it's something that as we generate more polygenic scores, we are indeed feeding into this thing that people are sort of taking and deploying in sort of dodgy ways, right? There's also the kind of feverish environment, shall we say, around how people think about their ethnicities, how they think about race. And so when you give human genetics to a racist, it inevitably reinforces what they think about their superiority, right? And that's certainly the case when it comes to these particular individuals who believe that the white race was genetically predetermined to rule and to consume milk because they have lactase persistence alleles at higher rates than everyone else, right? So they can consume milk like absolute idiots with their shirts off. So and there's a major subculture of absolute idiots on social media who it's kind of fun to kind of look through and sort of laugh at, but they're there. Many of them are real people, some of them not. But this sort of information does frequently reinforce their views, especially the stuff around educational attainment, income, that sort of thing. So that's why we work in disease. Oh, and just because I actually, this article made a real impact on me. New York Times 2018, Amy Harmon wrote about this particular phenomenon a long time ago now, actually, but it was an absolutely excellent article and opened up my eyes. I mentioned that polygenic scores are susceptible to biases in terms of ethnicity and ancestry. This is largely, as many people are probably aware, there's a lot of clinical tests and devices which suffer from this sort of a thing. Polygenic scores are susceptible to it because they're based on data which tracks very, very closely, population diversity around the globe, right? So if you develop a polygenic score, you need to use a multi-ancestry heterogeneous data set. Otherwise, it's not going to be representative. It's not going to generalize to other populations that you don't necessarily have as part of your training data. And that's absolutely the case because GWAS have been done predominantly a while ago, like 90% plus in European ancestries. So there's been, and you see, right? Well, and I should actually back up because it's not necessarily GWAS that is at fault here. The original cohorts have been European ancestry, largely European ancestry going back for decades and decades and decades before we had genotyping arrays, et cetera. So it was always baked in. It's come through now to human genetics and a lot of recent research, but including polygenic scores and other things. But going back to the root cause of the problem, which was our ability to, which populations we actually focused on for our research, and how much resources we put into recruiting people from diverse backgrounds, including different socioeconomic statuses. We're starting to make headway in terms of getting more diverse cohorts. For example, US All of Us, Our Future Health around here is making a special effort to do so, to increase the diversity of their cohort. So we're starting to make headway, but what we find now is that with polygenic scores, we have an inbuilt bias against non-European ancestries. We can do some things to address this, whether it's playing with doing things to the allele frequencies and the LD to try and account for that. But ultimately, we need more diverse data. You just can't get around it. It takes a lot of effort, but we're making some progress there. This originally was actually years ago. This was a bit of a gap, if not a risk, for me. But we've made some progress on it. So it used to be that the report, how people would report polygenic scores, was just like all over the shop. They would report different performance statistics. They would do analyses in really different ways. And many cases, roughly 40% of the time, they didn't actually provide the variance and the weights for the polygenic score. You'd have to either email them, they'd say yes or no. If they said no, they might be trying to commercialize it or whatever, right? So we found this, years ago to be a bit of an issue. You can probably see here's some examples of different studies and how they reported things. So we took it upon ourselves to start this thing called the Polygenic Score Catalog. You can go there at pgscatalog.org. Some of you probably have. And really, we did that because we wanted to standardize how polygenic scores were being reported. We wanted to make sure that people were making sort of comparable, interoperable performance metrics available, but also providing the polygenic scores themselves, because there was a risk years ago that the polygenic score field was going to sort of slide into this thing of not sharing the underlying scores, a lot of them being, not reproducible, essentially. So we started sort of small. It's gotten a lot bigger. Nowadays, the Polygenic Score Catalog has nearly 4,000 polygenic scores in it for over 600 traits, and we've curated over 500 polygenic score publications, which meet our entrance criteria, which include external validation and demonstration that the polygenic score has validity, right? So that's been, I think, useful for the field. Very much made possible by an amazing sort of postdoctoral fellow in the lab, now assistant professor Sam Lambert, and Jackie MacArthur at the EBI, who used to run the GWAS catalog, but is now at the BHF Data Science Center. And just to go real quickly, you can see this for yourself on the website. The polygenic score catalog includes a whole bunch of information around like what trait is the polygenic score trained on, the methodology, the underlying summary statistics. Now we include ancestry composition of those summary statistics and the score development samples, all the way through to how the actual association modeling is done, what covariates, what are the validation demographics, as well as critically the polygenic score file, which includes the SNPs, the risk alleles, and the weights. And we also bank the performance metrics, some generic performance metrics. And this really fed into a collaboration, a really excellent collaboration with the NIH's clinical genome project and their complex disease working group, which is led by Jen Wojcik there on the right, and on the left, Hannah Wand. And that I think really synergized well and sort of set the stage for constructing a set of reporting standards for the broader community for polygenic score studies, and that's something that we published a couple of years ago now, but has been taken up really widely and is now essentially not necessarily enforced, but very much encouraged by some journals, particularly Nature Springer. I think Cell Press does it as well. So hopefully you come across these things because it gives you a good framework for how you should sort of do and how you should report polygenic score studies. And I think what we've seen in the catalog is that it's sort of these two things have really worked together to really harmonize the field and open up a lot of polygenic scores, make them available, and has facilitated a lot of external, totally independent benchmarking studies of polygenic scores to more rigorously demonstrate their validity, and hopefully means that we get better trials out the other end. We also showed in the PGS catalog paper, again, the issue with transferability across ancestries. This in particular is looking at colorectal cancer polygenic scores. You can see that there. But it also shows that not all polygenic scores are created equal. And that's something that frequently gets lost when people talk about PGSs. They'll go and they'll construct their own PGS, and then they'll talk about all polygenic scores for a particular trait. When, as we all know, you really need to benchmark because some scores are better than others. And that's what a lot of the polygenic score catalog helps to facilitate. So that's one of the downstream things that we're working on, have been for a little while now, but it's taking a while to get together. And that's really taking the polygenic scores that have been deposited in the PGS catalog and benchmarking them systematically in external cohorts of diverse ancestries, and looking at the relative performances of polygenic scores in these external studies. And what you end up seeing is, you can look through and try and match polygenic score up a specific set of variants and weights to your particular clinical use case, right? Whether you're looking at a particular trait, whether you're looking at a particular ancestry, and hopefully that helps guide which polygenic score you might select to look at in your own data, potentially to run a trial on. And Sam's been leading this work with some other collaborators as well. And ultimately, we've made a nice little interface on the PGS catalog benchmarking, which allows people to sort of explore this benchmarking and interact with it, and hopefully find the polygenic score that works the best for their particular case. So this sort of brings me to a broader sort of framework, which I'll talk about just briefly. I mentioned the ICDA PRS task force. We were focused there on how do we actually use polygenic scores responsibly and equitably in healthcare, and to do that, Chris and I sort of brought together a really diverse cross set of people, whether it was geneticists, statisticians, but through to ethicists and lawyers and behavioral scientists, social scientists, that sort of thing, so that everyone was on the same calls, working on the same documents, making sure that they brought their particular perspective to ultimately to this paper, which I still see as the sort of benchmark in, like it says, the potential benefits, the risks, and the gaps for polygenic scores. It looked at it in a whole bunch of different ways. I thought it was pretty comprehensive and clear-eyed. And the only issue with it now is that it's a little bit old. So I would hope that someday we could bring this group back together and sort of update it. But again, I think this really highlights some of the things and goes really into depth on some of the issues that have been raised here, and brings up others as well. So a bit of a plug, but you check it out. Okay, so that sort of gives a firm base in terms of the genomics, right? The systems, or the genomics part of the systems genomics. We've been looking more recently at how we combine polygenic scores with other systems level data, other omics, for the purposes of risk prediction. And this is not a lone pursuit, shall we say. There are lots of people doing this, including people probably here. And in particular, looking at things like proteomics, so the UK Biobank's OLINK data, which just came out, the UK Biobank's Nightingale data on, I think, 300,000 people currently, expanding to 500,000 soon. So these multi-omic, very large data sets are broadly available now, right? So this is going to become a question of how do we actually combine these data to better capture disease risk. I was hoping to sort of dive into, I was sort of debated whether doing like a single paper talk here, just to kind of give you guys a deep dive, but I couldn't ignore like all the other stuff. So I've got like one slide on this cool paper that a senior research associate in the lab just put out on medRxiv, Scott Ritchie, where he was looking at the 300,000 individuals who have Nightingale data in UK Biobank right now, and was looking at to what extent could we actually combine polygenic scores, the same ones I mentioned before, with the Nightingale data to predict cardiovascular disease risk, over and above the current sort of state-of-the-art risk prediction model, which is SCORE2, which comes out of the unit that I'm in at Cambridge. So hopefully as relevant as we possibly can get. So we took the UK Biobank data and basically looked at the subset that had the relevant data, right? The conventional risk factors, metabolomics, polygenic scores, looked at the individuals who weren't already on statins, who didn't have prevalent cardiometabolic disease, and ended up with about 170,000 individuals and about 5,000 incident cardiovascular disease cases. And we looked at stuff, hopefully you can see the cursor. We looked at individual metabolites, both on Nightingale and actually on the biomarker panel, and their sort of incremental gain, at least in C index. And then we started combining these, the NMR ones into NMR scores, and looked at this usual stuff, right? Like hazard ratio, C index, delta C index, that sort of thing. And basically showed that when you add the PRSs or these NMR scores to SCORE2, you see sort of similar gains in terms of C index. But when you look at the net reclassification improvement, you see a little bit some separation now, where polygenic scores kind of give you a better sort of NRI for the cases, about the same for it doesn't make much difference for the non-cases, but for the NMR scores, the Nightingale scores, you actually get a greater improvement in terms of the case NRIs, but maybe a little bit worse in terms of the non-cases. So these two things together, and knowing that they provide largely orthogonal information, then gives us something really interesting to take forward into this population, this public health modeling framework that I showed you a little bit about before. So we went through the same thing, essentially simulating another sort of 100,000, simulating a population of 100,000 people, and looking at what would be the sort of change in the number of CVD events prevented, what would be the number needed to screen in order to prevent one event, what would be the number needed to treat in order to prevent one event, that sort of thing, and looking at what could be the potential public health benefit for using either individually or in combination polygenic scores and these NMR metabolomic scores. And ultimately what we found, I'll just sort of end this sort of story here, is that if you use score 2 only, you end up with about 201 events prevented per 100,000 screened per 10 years. You add in PRS, this number goes up to about 277. If you only add NMR scores, the number goes up to about 336. But if you add both, the number of CVD events prevented goes up to about 370. And that's all while keeping the number needed to treat in order to prevent one event somewhere in the range of 23 to 24. So fairly constant. So we sort of thought, this looks promising. It's not like game changing, but you are seeing a fairly appreciable increase in the number of events prevented if you were to be able to deploy these technologies at scale for the purposes of screening. The other thing I should mention actually is that this is focusing on this is a targeted approach. This is not doing PRSs and Nightingale for everyone in the population. This is only focusing on the intermediate risk group, where you've got 5 to 10% 10-year risk. Generally, actually, there are age and sex specific thresholds as part of score 2. But the point is that you're looking at a much lower number of people, while still being able to increase the number of events prevented. So that gives you sort of a taste of the potential of combining multi-omic technologies for risk prediction. We've also asked, to what extent can these polygenic scores, which capture population level disease risk, to what extent can they help you understand downstream biology? And to do that, again, we're integrating data across these different modalities, genomics, and in this case, proteomics. And to do that, we can do this study now, actually, in the UK Biobank with much greater power. But way back when, we were doing this in the interval study, which is, some people probably know, is a Cambridge-led blood donor study, where we could actually look at probably about 3,500 individuals who had Olink and somologic plasma proteomics measured and who had genomics available. And we could look at, things like, well, what's the relationship between polygenic risk of some cardiometabolic diseases, for example, coronary artery disease, and plasma protein levels. And then, what's the then effect of those protein levels on incident CAD or incident disease risk? And we could look at the correspondence between these two things, right? So here I'm plotting out. So this is a kind of a, I guess you could say it's a beta plot, where you're essentially comparing the hazard ratio to the beta estimate from a linear regression of PGS on protein levels. So you see this very close sort of clustering and positive relationship between proteins which have a positive sort of hazard ratio or positive effect on disease risk, and those PGSs for that same disease, which affect the protein levels, and vice versa, right? And you see that in particular for CAD and type 2 diabetes, but you see it in other diseases as well. So because we have individual level data, and I know this is sort of a territory that is very much of interest here because you're the Mendelian randomization experts, but because we had individual level data, we could actually run things like mediation analyses to look at, so we could try to estimate, what is the direct effect of this polygenic score on incident type 2 diabetes or coronary artery disease versus the indirect effect, the one that flows through a particular plasma protein, right? And you can also estimate, other stuff like, an effect through a confounder or that sort of a thing. So when we did that, you can sort of ask, well, what percentage of a polygenic scores association or with the outcome is mediated through a particular protein, and then ask whether that was significant or not. And when we did that, we found that, for coronary artery disease, a lot of the same sort of proteins which displayed this association with the PGS also showed a significant or, statistically significant mediation effect, and quite a few more with type 2 diabetes as well, including some pretty well-known ones, like insulin, what is that? Insulin growth factor binding protein, and sex hormone binding globulin there at the end. So just in the last few minutes, running a little bit behind, sorry. I just wanted to present the last sort of major work, or the most recent major work from our lab, which is trying to use genetics in order to capture variation and predict variation in multiomics, right? So we know that circulating molecules, all sorts of human traits, a fraction of that is heritable and is sort of to some extent predictable by the germline genome, right? That's the case for protein levels. That's the case for metabolites, et cetera. So why can't we use genetics in the same way that we're constructing polygenic scores to predict proteomic data, to predict transcriptomic, metabolomic data, et cetera? So that's a question that was essentially asked and to some extent answered for transcriptional data years and years ago by Heike Ehm and colleagues at the University of Chicago. And we want to kind of expand on that, build on that, sort of bring it into up to date with the latest technologies. I won't necessarily go over this. You probably know like some of the things you can do with predicted multiomic data, whether it's biomarkers, predictive modeling. You can look at disease subtypes, et cetera. We're particularly interested actually in decomposing protein variation into different components, whether it's G, E, and noise. And you can do things like look at reaction fluxes as well. So trying to look at, actual reaction rates, estimating those. And with a few tweaks, as you probably know, especially if you check out the trend signals, you can start thinking about using these sorts of things for these imputation models for causal inference. But I certainly acquiesce to the expertise here on that. This is just a little bit about the interval multiomics panel. It gives you a sense of the different platforms that are available, whether it's Olink, SomaLogic, Nightingale, obviously, metabolomics, and there's a few other platforms that we're working on as well, sort of Genos, which is glycomics, and CellCarta, which I think is different other proteins. And this paper came out a few months ago, led by Yu Xu, who's a senior research associate in the lab, using the interval multiomics data in order to train these genetic imputation models for Olink, SomaLogic, whole blood RNA-seq, Nightingale, and metabolomics. I won't sort of belabor how we did that, cross-validation, Bayesian ridge regression, et cetera. And whatever we're looking at here in terms of the interval performance is always in the test folds. We also did extensive external validation, which we really focused on from the very beginning, trying to get as diverse a cohorts as we could in order to test the transferability of these multiomic predictor, or these genetic predictors of multiomic data, working with sort of colleagues around the globe. But in particular, the Jackson Heart study, which is largely African Americans, as well as the Singaporean multi-ethnic cohort, which is the three main ethnicities in Singapore, sort of Chinese, Indians, and Malays. So across all these different modalities, we could construct a little over 17,000 multiomic scores, or scores that can predict multiomics data. And a little over 10,000 of those were Bonferroni significant in the external validation. And that sort of gives you a breakdown of how many scores were significant for which platforms. And just to give you a sense from the proteomics side, of the 2,300 or so scores that were developed, nearly 600 or so could, in the test sets, explain 10% or more of the variation in the directly measured protein levels. And about 143 of those proteins could explain over 30%. And a fraction of those as well could explain over 50%. Whereas in contrast, Olink, there was about 308 scores developed, and a little over 100 could explain 10%, 20%, and 20 could explain over 30% of the variation of the protein levels. just to update, we're continuously improving and enhancing the resource. So we've actually got the 3K Olink UK Biobank scores already developed, and hopefully are going to be releasing those in the coming weeks. And we've actually expanded the SomaLogic to the 5,000 SomaLogic platform currently. So, essentially more than doubling, 5xing and more than 10xing those proteomics platforms. So in terms of the predictability of each platform, not all platforms are created equal. You can probably see that, and these are not random selections of proteins, I should add, but Olink seem to be a bit more predictable than other platforms. Nightingale, particularly due because of to the correlation amongst the traits, very high correlation sometimes, had an odd distribution, but it sort of gives you a sense of which platforms give you what sort of performance in terms of R-squared. This is just saying that the vast majority of the scores are not just single QTLs, that they're actually quite relatively sort of distributed around the genome. At least, multiple LD blocks, that is, and especially within a single LD block, there are multiple conditionally independent variants for each score. So that was all nice. That was all within interval. In external validation, we found that the scores transferred expectantly relatively well to European ancestries. So interval to Finland, interval to Swedish ancestry, or interval to Orkney. There's some variation there in terms of the performance, particularly with respect to missingness rate of SNPs for the traits, but largely performance is maintained. But when you go to other ancestries, of course, you see this reduction in performance, right? So particularly for Nightingale, we saw a fair few scores becoming, quite non-predictive in Chinese, Indian, and Malay ancestries. But for SomaScan proteomics, we weren't able to measure Olink. SomaScan proteomics seemed to sort of hold up relatively well, where you could see that in sort of particularly in Indian ancestries, as well as Malay and Chinese. African American, you saw roughly half of the R-squared in external validation, as you did in the interval test sets. And of course, with these data, you can empirically look at which proteins in particular transfer well across which ancestries. And all that's available on the OmicsPred site. Just real quickly wrapping up, we've also done a phenome-wide association scan in UK Biobank, creating 500,000 proteomes, metabolomes, transcriptomes, et cetera, and doing association analysis with the disease outcomes. We find stuff that's both known and that's new, particularly validating some of the coronary artery disease signals from the Ritchie et al. Paper that I presented. So most of those showed really highly significant associations with incident myocardial infarction in UK Biobank. And like I mentioned, we're going to be releasing a new generation based on the UK Biobank Pharma Proteomics project and the Nightingale data as well. There's a little bit of biology around the JAK-STAT signaling pathway, a component of which is IL-6, and which is probably of interest here. So you can use these scores to understand and identify sort of known biology and new and interesting drug targets. And we've made this into a portal that is open, and you can go and you can download all these scores for free and use them for both academic and industry pursuits if you so choose. And this website's also going to be, this resource is also under development. We're going to be moving it to Google Cloud, so there's actually going to be a backend to it at some point, and hopefully a next generation sort of resource that, in my dream, sort of would start approaching the PGS catalog, but we'll see. Okay, cool. So I'll just end with some high-level takeaways that we've really, since this all started like over a decade ago, we've come a long way, and there's still, we're only really scratching the surface in terms of what systems genomics or multi-omics is able to do. The polygenic score research has really improved dramatically, thanks to things like openness standards and addressing some of those risks. And even so, PGSs have clinical potential, but, and you saw it, we need the translational studies, we need the pragmatic trials. We actually need to start measuring whether they have clinical utility in particular pathways or not. Polygenic scores, despite that, can still be used to understand disease etiology, as I've shown in a few ways. And ultimately, you sort of start thinking about, well, to what extent can these polygenic scores, are they offering some sort of genetics landscape, right, which constrains or regulates a lot of the trait space or the multi-omic spaces that we're integrating with these data. So how we actually analyze, in a way, it's kind of like you start thinking about the frameworks and how in which you analyze multi-omic data and how that can improve both the disease risk prediction and how we can leverage it to extract etiological insights. So kind of changing this diagram, instead of like really level sort of sheets and more sort of complex landscapes where the transcriptome sort of molds itself to the genetics, and maybe a little bit less on the proteomic side and less so on the metabolites, microbiome, et cetera, but really trying to understand what are the relationships again between these different levels and how we can leverage them. And I've sort of mentioned them throughout, but these are the actual heroes on the ground who are doing the real science, thankfully. Some in particular people on the polygenic score side: Sam Lambert, Gad Abraham, and on the multi-omic side, really, Scott Ritchie, Yushu, Yang Liu, and co. So that's the lab, and that's it.
Amazing amount in there. Questions?
One obvious question that I had about comparing between polygenic riskers between ancestries. Have you ever compared like immigrants versus non-migrants? Because in a lot of your papers, I see African Americans, but I don't see Africans in Uganda. I noticed you brought this into, so Uganda, so
Yeah, so migration. Yeah, it's a great question. It's one that you really wonder about in terms of gene by environment interactions and that sort of thing. So lots of ideas, lots of aspirations in that space, but we don't have good data. The migrational patterns are unfortunately intertwined with population structures and whole communities moving from A to B, and then having to kind of compare population structure within a country to the population structure that migrated for some reason to another country. So that's a long-winded way of saying it's an important topic. There's a lot of cool stuff in there, but it's a really thorny issue to get at, how polygenic score, how migration and different environments can affect polygenic score performance, right?
Yeah, thanks for a great talk. I was wondering, you're talking about how the polygenic risk scores, they were not as good in other populations that were not the European populations. So would it be better to try to make bigger cohorts with many different ethnicities and just have like one kind of result that you can use for all the different ethnicities, or would it be better to have a lot of separate cohorts where you have like, so you use one score if you're European, another score if you're Asian?
Yeah, I mean, that's a question that a lot of people are thinking about. It to some extent, it kind of depends on who you talk to, what they think the answer might be. My preference is to have one score which transfers across multiple different, ideally all, but multiple different ancestries, right? You don't really want to get into the situation where you've got ancestry A, ancestry B, ancestry C, and you've got different scores for everyone, and then you have to bin people. And then you have people who are admixed, like me, who are just like, well, okay, what bin do you fall under? None. What's your score? none, you know? So that would be my sort of preference for those reasons in particular, having one score for everyone that maximally transfers.
Oh, like a really nice presentation. So the OmicsPred resources are really nice. The resource I think, but it's built on sort of helpful normal populations. So in the context of sort of identifying targets that impact on the progression of disease, ideally what you would have is a way of predicting the levels of omics within disease cohorts. So are you involved in sort of any efforts to develop polygenic scores in those contexts?
Yeah, so I mean, it's not so you would think that that would sort of be a limitation. What we kind of find is that, well, the genetic effects on the protein levels aren't modified by disease status. We largely don't find that, and I think it's because we find so many disease associations. I mean, maybe it's the massive sample size we're imputing into, but we find so many, and much of it recapitulates known biology, that it kind of makes me a little bit kind of, I mean, there might be some dilution of power there, but in the sample sizes that you're able to impute into, it's not really a limitation in practice. But yeah, but I would assume that if you went in and did proteomics in you know, case control cohorts, and were able to create, you know, to actually encapsulate, you know, variation of the protein levels that's associated with disease, you know, by definition, then you could improve your power further. So yeah, we're focusing on population-based to begin with, but I imagine there would be additional power on offer with by looking at cases. Yeah, specific cases.
May I ask how good is PRS in order to predict disease with a strong association, immune-mediated disease with a stronger association in HLA region? So in comparison.
Oh yeah, really well, actually. If I'm understanding your question correctly, how do PRSs perform in immune diseases with large HLA signals? Yeah, I mean that's kind of like
and
the.
Sorry, and the second part is in your answer based on what Liv asked previously in different population. I might guess that in this case you need to explore this in separate population, separate ethnicities, as HLA region can be transmitted quite differently across different people from different ancestry groups. There are different observed associations. So
yeah. Oh yeah. No, I mean you're kind of asking about the Bermuda Triangle of the genome, and like certainly as you go across different ancestries, the LD patterns change, the allele frequencies can sort of ceiling and floor and all this stuff. But despite all of that, you see, I mean, so celiac disease, type 1 diabetes, ankylosing spondylitis, and you know, various ones. There are these, you know, massive HLA signals, and that actually in many cases gives you you know, AUCs, right, that are approaching 0.9, sometimes exceeding 0.9, just based on the genetics alone. That's the case for type 1 diabetes, that's the case for type 1 for celiac disease and ANKSPOND as well. So if you got an HLA signal, you know, your polygenic score is probably going to be pretty good. And so a lot of the autoimmune domains, people are looking at combining polygenic scores with autoantibody screening to better diagnose cases. And if you have a therapy which can delay onset of that disease, then you're really talking. So just, I think last year, FDA approved a type 1 diabetes therapy, which actually delays disease for at least 12 months or something like 12 months or something like that. So pairing that with a PRS and autoantibody screening is a really exciting way of trying to catch these cases early and get them the therapy early. those particular polygenic scores, T1D and celiac, they transfer really well, actually, across ancestries. So if you look at South Asians and East Asians in particular, I know that's the case. You might have to correct for some allele frequency differences, but I don't know about the other ones, but they certainly need to be empirically tested to determine whether they do transfer. I would assume that the whole HLA signal doesn't disappear, because if it did, then, well, I'd assume the actual prevalence of the disease would be a lot less in that particular population. But, yeah, hopefully that answered your question. Yeah.
Well, we'll thank Mike again. And, he's around all of them, but quite busy.
Yeah. Thank you.
Yeah, I'll talk to you about the chat. Upload slide later sometime.
Yeah.
Over beer.